Ruben asked me to speak today about nutrient density as a sort of part of today's presentation. It's a word that I've helped to coin, and I think you could even begin to call it a movement now. It's starting to coalesce. So maybe I'll just give a couple background context framing things, and I want to show some slides and what is it and things like that. So this is just a picture of the farm I grew up on, which is about 20 minutes north of here, which is Many Hands Organic Farm, where NOFA actually was housed. NOFA Mass was housed for about 35 years. So I grew up not only on an organic farm, but my parents ran an organic farming organization. So I come to the whole conversation with that context. I like to say it was only when I got married and had to figure out how to make a living that I realized I wasn't a good farmer. I had been farming all my life through my teens, through my 20s. I managed my parents' farm in the summertime and travel the world in the wintertime. But I didn't need much money to travel the world with a backpack, right? $5 a day in India is tons of money, right? You don't need a hell of a lot. So I wasn't living on much. But when I got married and I had no other viable skill sets besides farming for making a living, is when I realized that, if I didn't have pests and diseases, maybe I would have more economic viability. And I had been normalized. Pests and diseases were normalized in my childhood. It was just sort of the way it was. Potato bugs ate your potatoes, and powdery mildew took out your squash and your cucumbers, and flea beetles took out the brassicas. I mean, everybody knows the general pests and diseases. What I didn't realize until I started doing some research was that those are symptoms of poor health. You would argue if you have a chicken being eaten alive by a flesh-eating fungus, like a staph infection, you would say something's wrong with that chicken, and you probably wouldn't slaughter that chicken and eat it. But if you've got a squash plant being eaten alive by a flesh-eating fungus, which is called powdery mildew, which happens to almost all of our squash plants every year, we happily harvest a squash and eat it. If you had a cow that had flies had laid eggs on its hind end, and the eggs had hatched, and those maggots eating a cow alive, right? Sort of viscerally being eaten alive by larvae for animals is a sign of something really being wrong. It actually is also a sign for things being wrong in plants, and there's a whole a science where we can understand this, and the reality ends up being that when you are managing your soil in such a fashion as the plants have access to what they need, they actually physiologically have insect and disease resistance. They become indigestible to those pathogens. And it was when I started to change my practices, still staying within the organic rubric, but get these kind of results, that I got very excited. A, because I was able to make my living working 20 hours a week, but B, because I thought there were some very exciting implications here, connecting soil health and farm viability and human health, etc. So we coined this word nutrient density to refer to the variation in nutrient levels in food. People, I'm sure, have had the experience of eating a carrot that didn't particularly taste pleasurable, and then also having carrots that did quite taste good, or peaches, or strawberries. That flavor variation is a nutritional variation. We are actually wired with very, very sophisticated nutrient monitoring devices called noses and tongues. I think something like 30% of the human DNA is associated with the function of your nose and tongue. Like, nature thinks it's really important for you to smell and taste well, because what you ingest becomes who you are. And if you ingest insufficient or improper sustenance, you begin to degenerate. There's a direct connection there between what you eat and your physiological, psychological, emotional health. It's really quite exciting. Started giving workshops and courses in 2008 or 9, I believe it was. Founded the BFA in 2010 as an organization to focus on this question of nutrient density. Workshops, courses, conferences sort of spread out from here in New England, around the country, into other countries, and then into other continents. As we taught these courses, we found people were getting positive results across the board. It didn't matter what the scale was, it didn't matter what the geography was, it didn't matter what the crop was. The more well you work with soil life, the healthier your plants become, the better your economic viability becomes. soil health, flavor, aroma, shelf life, etc. I'll just start running through some slides here to sort of, and then we started doing research in 2016, 17. So that's what I want to focus on here. BFA, founded in 2010. Our mission is to increase quality of the food supply. By quality, we're talking about flavor and aroma, nutritional value. We established this term, nutrient density. I think I just said this. Conferences, courses. People may recognize some faces here. These are people, some of the people we've had speak at our conference over the past 10 years. We actually have a conference going on right now, online, 16 weeks globally. We have over 150 people from Africa attending, and 70 farmers from Ireland. It's a really massive global event, very exciting, talking about this topic. And I think you said you were attending. Yeah, you're beautiful. Brilliant. So, 2017, we started this work with the research. The thought was, could you, I guess there were a couple questions. The first question is, how significant is nutrient variation in food? Our thesis was that it was significant, but there didn't seem to be sufficient data to verify that point. Our second question was, if there is significant variation in nutrient levels in food, what are the causal factors? Does it connect to soil type or seed or variety type, or is it certain practices, or is it soil health? If there is variation in nutrient levels in food and it's significant, what causes it? Our thesis was that there was significant nutrient variation and that it connected to soil health, not to variety and soil type. And the third question was, is it possible to build a handheld meter that could be used to assess that nutrient level in food in real time? So we're talking basically a tricorder from Star Trek. Like, you walk up to the carrot, flash a light at it, and it spits out a reading, 20 out of 100 or 80 out of 100. The concept is there's a scale. This is the vision, or the thesis, is that there's a scale of nutritional level in food, and some stuff is 20 out of 100, and some's 40 out of 100, and some's 80 out of 100. And our thought was, if we could support people who buy food in being able to choose between the three different types of carrots on the supermarket shelf, or the farmer's market, or whatever, if they could know that one was 20, one was 40, one was 80, they might choose the 80 and leave the 20. And if that was the case, we could begin to apply an economic incentive to agriculture to focus on nutrition as opposed to volume. Because right now, agriculture is primarily focused on a volume as its economic incentive framework. You get paid by the pound or by the bushel. You don't get paid by the flavor, right? So we started this process. In 2017, we built our first handheld spectrometer. We built one and said, okay, look, it can be done, for those who said it wasn't possible, to build a handheld consumer price meter. I've got a second generation one here that you can see. It's nothing too fancy. It's 3D printed, but this is, we'll talk about what it can do in a minute. So 2017, we built our first meter. 2018, we built our first lab. We had people send in carrots and spinach. Is that a question? No. Okay. Spotting flies. From grocery stores, from farmers markets, from farms, organic, not organic, Michigan, Maine, Minnesota. We wanted to survey the supply chain. So that was just year one, was just carrots and spinach. Year two, we set up our second lab at Chico State in California, and we started looking at four more crops. I think it was cherry tomatoes and lettuce and kale and grapes. And we had farmers send in the soil those crops grew in, along with management data. Give us all the, did you till? If so, how deeply? With what? How many times? Did you, fertigate? What was in there? Did you put cover crops down? What was the mix? When did you plant? How did you terminate? What was the variety of the seed? Just as many management questions as possible. And then when they picked the 3 carrots out of the soil for triplicate samples, they would pull the soil at multiple depths underneath each carrot, so we could actually overlay soil metrics against nutrient levels, against environmental conditions, causal factors. 2020, I think I've got a slide here for the 2020 data here. You can see we set up our third lab in Europe. We had, I think it was over 200 farms that year who participated. Every one of these green dots is a farm Green is farms that participated. Orange is citizen scientists that were buying at stores and sending samples in. You can see the number of crops we worked with that year, the total here. So we have a little over 10,000 samples right now in this database of crops from around just North America and Europe. But it's many, it's a lot. So we can actually begin to build a significant data set. In our initial work, we were only looking at a few different compounds, so antioxidants, polyphenols, and some elements in the crops, and then the soil. We measured these things. Research is expensive, and science takes money. We're a charitable, nonprofit educational organization, so we're running on donations. So we couldn't, at least initially, do the level of nuance of science that we wanted to do, but this is a good baselining process. So what I wanted to share here is just some of the variations we found. I'll just some of the basic results. If you look here at, so these are all just carrots. We have data like this for over 20 different crops. So this was a sulfur, and the lowest is 8.41 milligrams per 100 grams, and the highest is 33.19. So 33 divided by 8, you get about 4. Anyway, so this is a 4x variation, as in like this carrot has 4 times as much sulfur as that carrot. And what I want to emphasize here is these are the color ranges, or the fifths of the range. So this is 0 out of 100. This is 20, 40, 60, 80, and 100. So if anybody ever went to, say school and had a final grade at the end of the year was a 58. When I went to school, that was an F, right? That would be a failure. So is it? I'm not sure. They've changed schools, like all kinds of things are. It's a downward slope, but anyway. So if this is 60 out of 100 right here, and that's 58 out of 100, the vast majority of the samples are below the 58th percentile. Actually, the peak of the samples, the most samples are in the 25th percentile of what's possible. Not only is there a 4x variation here, but the vast majority of the supply chain is sitting below 50. Below half of what's possible, right? Almost everything is relatively poor. We can see a couple farmers are doing a good job. Some farmers are able to get high levels of nutrients in their crops, but almost no one. Almost everybody's doing a bad job in this element. Phosphorus is another element, 11.7 to 93.69. That's about a 8x variation or more. So this carrot has 8 times as much, or these 2 or 3 carrots have 8 times as much sulfur as those ones over there. Again, peak of the graph is in the 25th or 30th percentile. So the vast majority of the samples we've found are very low in relation to what they could be. And the variation is not small. It's not 2% or 4%. It's 8x. This is data that just follows with all the crops we've got. Here we have polyphenols. Polyphenols are a flavor, aromatic, sort of health-giving, anti-cancer, chronic disease kind of compound. 2.34 to, you can't see, it's 49 point something over here. That's a 20x variation. 20x. This one has 20 times as much as that. And the peak of the graph is in the 10th percentile. And then over here, antioxidants. Another one of those health-giving compounds. 4.92 to 195. That's a 40x variation. There are literally 40 times as many antioxidants in these carrots over here as there are these carrots over here. Peak of the graph is, this is 20, right? So it's around the 20th percentile. So the vast majority of the carrots in the supply chain are relatively quite poor in relation to what they could be. And I'll just say, as I said before, if we have time, I can show you the graphs for all the other crops. This is across the board, across North America, across Europe. We can presume, I think with some decent confidence, that this is a reasonable assessment of the supply chain. And for anybody who knows about bell curves, these are actually nice bell curves, right? They just have long tails. So if you're into science and statistics and things like that, you would assume that you'll find these representations in a good data set. It's not evenly balanced. Like, it's just got a long tail. So we can see that some people are doing a relatively quite good job in relation to others, but almost no one is. I made that point about 5 times. That's probably sufficient. Okay, so what causes it? Here's variety. There's Napoli, Bolero, Nantes, Mokum, Romancea. You can take a Napoli seed and produce a carrot which is relatively quite low, and you can take a Napoli seed, produce a carrot which is decent. Right? Bolero, same thing. You can get a low quality carrot. This is an average. BQI is an average of 6 elements and 2 compounds. So copper and zinc and calcium and phosphorus and potassium and sulfur and polyphenols and antioxidants. We just averaged them all up together to get this sort of range. So I'm not seeing any connection to variety here. I'm not seeing any variety that's like, oh, when you plant this variety, things are good. And when you plant that variety, things are bad. What I'm saying is, you can do a good job with any variety, and you can do a bad job with any variety. So that would argue that variety does not connect to nutrient density. Right? It's not a causal factor. The specific variety of the crop is not a causal factor. Otherwise, we'd see some kind of pattern different than a overall representation. This is north. This is the by USDA climate region. So there's 7 different climate regions according to the USDA: Northeast, East, North, Central, Southeast, Central, Northwest, West, Southwest. Again, I mean, we're based obviously in New England, and so we have more relationships up here. So we have more samples from here. These are from where we knew where the crop was produced. This is from farms. But yeah, just because you are a local Northeast farmer who may be organic and is practicing no-till does not mean you're doing a good job. That's who most of these people are people in our circuit of influence, right? East North Central. So you can see no connections. I don't see any connections between soil type or climate zone. Yeah. Well, some things do break down. So polyphenols and antioxidants will break down over time. Things like copper and zinc and sulfur are elements, and they don't leave. I mean, even when that squash is totally rotten, the copper is still there. It doesn't go away. So, yeah, there is definitely a degradation over time. Our question was, what is the nature of the food that's on the shelf? We were not trying to tease out all the background. How long did it ship? How local was it? We would presume we got some decent confidence that when something's fresh, like all other things being equal, something that's picked fresh is going to have higher nutrients in it than something that's a week old or 10 weeks or 2 weeks old or 4 weeks old. But the reality is, if you got a 40x variation, most of the crops are picked. I mean, even the fresh ones, right? It was a 2 days or 3 days. A lot of these samples are from farms who picked and shipped. But yeah. So by sample source, here is from farms. This is from farms, direct from farms. Not a lot of greens, so a bunch of reds and oranges. This is from the grocery stores. Doesn't look a hell of a lot different. Here's the garden, and here's the farmer's market. I don't see any dark greens from some garden sources or farmer's market sources, but I do see some dark greens from the grocery store. We see across the board similarly mediocre levels of food nutrient density in all points of purchase. This is not what small farming people want the answer to be, right? But we've got lots of samples from lots of crops from lots of years from lots of countries. Over here is by practice. Here's certified organic. Here's people using cover crops. Here's no-till. Here's tillage. No-till and till. Here's organic, not certified. Here's reference and none. Those are grocery store, not organic. Right there. Grocery store, not organic. It's right here. Like this is fatter farther over than there, right? This is backyard, not certified, small organic is right here. Like it's fatter right here. That means that's what the most samples are. Over here is grocery store, not organic. I'm sorry. This is not. It's not what some people want the answers to be, but like, it is the reality. Okay, here is regenerative. Here is biodynamic. There's local. There's greenhouse, light till, and heavy till. So, yeah. So, across the board, we're seeing variation. Now, we're not seeing that it's the variety. We're not seeing it's the soil type. We're not seeing it's the point of purchase. We're not seeing it's any one individual certification label or practice. Right? The slide I don't have here, which I should have, is a slide which shows that it's the level of soil respiration, the level of life in the soil, is the only thing that we found that connected to nutrient levels. The more the life in the soil breathed, the higher the nutrient levels were. Like the more people down there breathing, right? Like each microbe emits, or not all of them, but a lot of them are aerobic. They emit carbon dioxide. And so we did a really simple test. We put soil in a box with some water and a little carbon dioxide meter. And we tested how much carbon dioxide came out of that soil in 12 and 24 hours. It's called a respiration test. Really, really simple. And that was the only thing that we saw that connected to nutrient levels, right? Which to me is extremely exciting, which actually verifies the thesis that we had, which is that it actually aligns with a lot of things. Like we understand from evolutionary biology that the plants were evolved by the microbes. Let me know this story. 420 million years ago when there were no plants on land, it was the microbes that evolved the plants to make sugar to feed them, right? I mean, people know this, yes? So, like, I mean, what I've been teaching for many years, so I teach courses, is it's not about the crop. I don't care whether it's a carrot or a potato or an apple tree or hay. It's about the soil life. Only when you create a dynamic where the soil life is functioning well should you expect the crop to function well, et cetera, et cetera. So I just don't have that graph. I'll just say the conclusion of all this work. We had 3 questions: do nutrients vary dramatically? And it sure looks like they vary dramatically. And is it soil health that is causal, or is it soil type, or variety, or no-till, or whatever? And it sure looks like it's the soil health that's the causal factor. And then the third question was, is it possible to build a handheld, consumer-priced, non-invasive meter that can be used at point of purchase to assess nutrient levels? And that's what we did in 2021, was we released this meter. Just a few hundred of them around the world, not too many, because we weren't making any money on them, but we wanted to make sure it could be done. So we built a bunch of them, and they all correlated with each other. You flush one at a kale, and it gives you the same reading as you flash another one at a kale. Just doing that is actually a really, really pain in the butt. I'm not sure if anybody's ever tried to build scientific instrumentation before, but it's not, much less write apps and things like that. So anyway, for 10 crops, this meter is calibrated to 1 or more compounds. So I think it's antioxidants in carrots. We have a statistically significant data set, which is calibrated to the meter. So if you flash a light at a carrot, it'll tell you this carrot, the antioxidant levels in the 20th percentile or the 40th percentile. It's not telling you the overall quality of a carrot. It's just telling you antioxidant. With zucchini, it's polyphenols. We only have a statistically significant data set with an R-squared above 0.8 or whatever it is. That everybody knows statistics, know what an R-squared is. Like, we have to get an R-squared above a certain number, which gives you the confidence to say this is a trusted, a trustable algorithm. So we have, for zucchini, it's like I said, polyphenols. So that was the end of phase 1. Phase 1 was, do nutrients vary dramatically? If they do they connect to soil health? If that's true, is it possible to test nutrient variation at point of purchase? Because if all 3 of those questions can be answered, yes, then our thought is we can disrupt agriculture in a very meaningful way by providing people the ability to test food before they purchase it and letting the market sort out what people want and don't want. So that's where we started working on our next level of it, which is actually trying to define nutrient density. This is the beef project, which is just about complete this year. Our thesis is that what the animals eat connects to the health of the meat that you eat from them, which probably is not too radical of a concept, but like cows that eat what cows used to eat in the olden days have beef that is better for you, and cows that eat corn have beef that's not so good for you. And so, this slide is quite vague. So what we're doing is it's a global project. Farmers are sending in a steak from 3 different animals. Each steak, we looked at about 200 different nutrients in that steak. So elements and vitamins and enzymes and amino acids and fatty acids and proteins and lipids and terpenoids and phenolics. So hundreds of different nutrients as opposed to just the 10 or so we looked at initially. A bunch of different families of compounds. As well, we're having the farmers send in the manure from the animals so we can do a microbiome assessment on the animals. So we're looking at a couple hundred different species of microbes in the manure in different kingdoms, archaea and bacteria, looking at levels, not just the presence of the species, but the concentration of the species, because anybody who knows about gut flora knows that gut dysbiosis and healthy gut flora, what's going on in your belly really connects to how healthy you are. And so we want to overlay the nutrients in the meat against the microbiome of the animal. As well, we're looking at the forage or the fodder that they ate, so that people are sending that in. The soil that fodder grew in, we're looking at. The management practices of how those animals were raised, whether they moved every day, whether they, what was the nature of the scale, and how old were they, and what was their variety. And finally, I don't have it on here, there's a subset of this beef that has been fed to humans. And so we're doing human trials, short-term human trials, just like eat this meat, and then we'll sample your urine and blood in 1, 3, and 5 hours to see if we've shifted, if inflammation markers have shifted, etc. So they're getting the grass-fed meat and the corn-fed meat and the Impossible Burger, so we can see all those. So this is the process. It's about $2,500 per farm to do the assessment. Big complex job. And we're at the point now where we actually finally have all the data collected, and we're starting to write papers on this. We've got one coming out this summer that will be the first paper from this. But the idea where we're at now, well, I'll just go a couple more slides, and then we can talk about where we're at now. Obviously, primarily North America, definitely all different kinds of climate zones. You can see from, Alberta, northern Alberta, to the Gulf Coast and Texas, Hawaii, Australia, South America, a bunch in the UK. So we got samples from all over the world, all kinds of different climate zones, different varieties of animal, different age, etc. Grass-fed, corn-fed. So until we have everything published, what we're looking at, and we've got actually a comprehensive definition of nutrient density in beef. We're just looking at the omega-6 omega-3 ratio as a biomarker to say, what are we finding so far? Because, like I said, this is a project that's still in work, so we can't say completely what the answer is. But people here familiar with the omega-6 omega-3 ratio? Everybody know about omega-6? No. So this is a really important point. And I'm not as well versed in the specifics as I would like to be, but I think I understand the basic broad strokes. Omega-6 and omega-3 are different kinds of fat. You may have heard about saturated fat and unsaturated fat and have you heard about these different kinds of fat out there and not really able to track which ones are the right ones and which ones are the bad ones? And I haven't been able to actually track them all down, but there's a sort of a rough bifurcation. In a plant, you will have high levels of omega-3 in the growing green phase of the plant. That fat is in that omega-3 structure, and when it makes a seed, it stores the fat for the next generation in a different structure called omega-6. So they're both different kinds of fat. But what happens when if you eat too much of omega-6, then you get high levels of what's called inflammation, which is the root of chronic disease. People have heard about cancer and diabetes and osteoporosis and all these kinds of things, these chronic diseases that are generally managed by our current healthcare system. They're not cured, they're managed. At the root of chronic disease is this thing called inflammation, and at the root of inflammation is, in many cases, this high levels of omega-6. So it's a really systemic issue that is going on in the Western diet for the last 150 years. We've seen these shifts towards different diets for our animals, and people now eat a lot of seed oil, like margarines and spreads and things like that. Those are from seeds that have high levels of omega-6. So you're overdosing your body on omega-6 when you eat that. When you have a fish and chips or French fries, if there's anything that's fried in seed oil will have high levels of omega-6 in it, which is not what people used to fry things in. They used to fry things in lard, and they cook things in butter. And when you have pigs and cows that are eating green things, not grain, then their fat has high levels of omega-3 in it. So this is one of those really important differentiating markers in telling whether an animal product is good for you or not. Is this omega-6 omega-3 ratio? Very uncomfortable for the small organic farms that I grew up on. If you're feeding your chickens, your meat birds and your layers, and your pigs grain, doesn't matter if it's organic grain, doesn't matter if they're outside on grass, if the vast majority of their diet is grain, you have certified organic, very expensive, high omega-6, omega-3, high omega-6 animal products. Those are not good for you. Really, really uncomfortable information. Maybe less bad for you than the conventional stuff, because you don't have the glyphosate, you don't have the antibiotics and all kinds of other things in it. But from a nutritional standpoint, animals have not been eating grain until very recently, right, in history, the last 100 years, ever since agribusiness, changed things around, and grain became a commodity. But chickens didn't have their major diet as grain 150 years ago, right? Pigs definitely did not have grain as their major component of their diet. So anyway, we're just talking about cows right now. We can see across the board, regardless of geography, regardless of climate zone, regardless of age of animal, regardless of gender, of variety of animal, all the farmers that were doing a really good job, like with that running animals around on pasture, they're totally tightly packed right here. Right? This is they're all there. We have a few outliers. Those outliers come from, in this case, this was during COVID, a very large company which is a sort of an online, you order the meat and it comes to you in the mail company, whose name I'm not going to give you, was historically gets their beef from Australia because it's cheaper from Australia than it is from the US. And so they had a boat coming from Australia with a bunch of meat on it that was stuck off the coast of Southern California because the dock workers were overwhelmed and COVID was happening. And so this particular company, instead of going with the normal meat they get, bought some meat from the supply chain in the U.S. from a producer they didn't know, who claimed that meat was grass-fed. And technically, almost all cows eat grass at some point in their life. So you can legally say on a package of meat, grass-fed, with pretty much any cow, any beef, right? 100% grass-finished, that's a different statement. Pasture finished, just because the cows were out on a pasture at the end of their life does not mean you weren't dumping grain onto the pasture for them to eat.
Doesn't mean there's grass in the pasture either.
Exactly. So we've got all kinds of labels and claims that are there to connote a certain thing, but if you actually read the words closely with a bit of a cynical or skeptical eye, you may find justification for that cynicism or skepticism. So, at any rate, people can claim this is grass-fed. When they fill out the form, the submission form, they said this was grass-fed beef, but we knew where it came from, and we called them back. We said, look, we got 300 dots right here, and we got 8 dots right here. What's going on? And they said, well, this is what happened. So, anyway, the average here is 1.7 to 1. The range was from 0.9 to 11.4. Obviously, these are mostly outliers. 1 to 1 is considered to be like the holy grail of food. You want to have a diet at a 1 to 1 balance in your. People maybe heard about salmon as something that has high levels of omega-3, and that's why and fish in general. In nature, fish are eating phytoplankton, which is green, right? In farmed situations, like farmed fish, they're feeding them green. So your salmon doesn't have the your farmed salmon doesn't have the omega-3 omega-6 ratio that the wild-caught salmon does. In the same way that anyway, so as an outlier, as a sort of I'm digressing a little bit, but here's the range we found for the grass-fed. Here's the range we found for the grain-finished. Obviously, a much larger range here in grain-finished, based on the fact that not everybody finishes their animals the same. Some people finish with all grain. Some finish with a mix of hay and grain, etc., or all kinds of other agricultural byproducts. You'd be amazed to know what's fed to cows and feedlots. The range was 7.8. The average was 7.8 to 1. The range was 3.5 to 14.4. American Heart Association says anything more than 4 to 1 is bad for you. Anything more than 4 to 1 is bad for you. And why they recommend seed oils, I can't possibly understand, because seed oils are like 25, 30, 50 to 1. And yet the American Heart Association recommends seed oils. But anyway, more than 4 to 1, the literature's done, is not good for you. Below 2 to 1 is where we're looking for. And so we basically found a couple grain finished that were at 3.5. The reality is on your pigs and your chickens, even your organic ones that you're spending, moving around on the chicken tractors every day, 15 to 1, 20 to 1 is where your organic grain-fed chickens and pigs are, which is not good. Anyway, last slide here. The only thing we found that connects to, the thing that closest connects to that omega-3 omega-6 ratio is the number of species they have access to. The number of the diversity of the ecosystem connects to the health of the meat, which again was our thesis, and seems to be borne out by this data. So, with the project with beef, I'm using omega-6 omega-3 as a stand-in, but the objective, the conclusion of the beef project, is to hand the microbiome data to the microbiologists, the biochemistry data to the biochemists, the nutrition data to the nutritionists, the agronomy data to the agronomists, and have them say, this cow had a microbiome I think is in the 90th percentile. This cow had a microbiome I think is in the 10th percentile. So the microbiologists score the microbiome data, the biochemists score the biochemistry data, and then we overlap them and say, do the biochemists think the same cow is good and bad that the microbiologists and the agronomists and the nutritionists do? And if all those different fields of study come up with the same answer, then we'll find 5 biomarkers. What are the key nutrients in the meat that correlate with that range? And then we can engineer a next generation meter to test that. And that's where we're at right now, is working on building the next generation meter for animal products, which will plausibly be in the world next year, where, like I said, about all the organic pork and poultry that's been fed grain that isn't that good for you, like people will actually be able to go out and test animal products broadly and see what is and what is not good in the supply chain. So the opportunity to powerfully shift this, if we have the work from Ken Hamilton, this guy with the sprouting and the fermenting, on one side, and we've got the meter on the other side, I call it the carrot and the stick. Like, at some point, people are going to be able to figure out what's good and what's not, and they're going to go to their farmer they thought they trusted because their farmer is a friendly person and they don't know anything about farming. So they went to the farmer because that was a nice person, and they buy from them thinking that what they're producing is good. And they're going to go to that farmer and say, you may be a nice person, but your pork has omega-6 and omega-3, it's 25-1, and I'm not going to buy it unless you can get it down below 2. So that's the idea here, is we use that empowered, enlightened self-interest to facilitate the transition. And being able to do it on scale globally is basically at our fingertips. This is what I've been teaching around the world for the last 15 years. It's like some really rudimentary things that must be in place. You must have air in the soil for the microbes to breathe. If the soil's tight, there's no air, and they're dead. Same as a chicken, same as a child. Put a plastic bag over its head, it's going to be dead. When your soil's tight, microbes are dead. So air is critical. Water, again, really, really necessary. You don't water a cow for a week, it's going to be a dead cow. Right? So dry soil is anathema to life. There must be food for the microbes. So that's the organic matter. That's the cover. That's the the sugars coming from the leaves. Food, air, water. Like, these are not complex things, but in many cases people completely overlook them. A penetrometer is a great way to see if you've got soil compaction or not. Stick your hand in. Pick up a handful of soil. Does it feel powdery? Like, these things. So, and then the other ones are minerals and microbes. Like, you need some basic minerals. You need like the copper and the zinc and the boron and chromium and molybdenum, and you need a suite of microbes. So, in my experience, if you have all 5 things present, nature will basically do the work. Indigenous cultures were able to manage continent scale. It's agriculture, but it's way more sophisticated than what Native Americans were doing here when the white man first came. They were managing the whole continent proactively with a light touch, with no beast of burden, no wheels, no PhDs, no fertilizers, right? Because nature has everything we need. But the question is, do we know how to work with nature and facilitate that? So I think in many cases we get stuck on the science, we get stuck on the techniques and the theory and the measuring this and the measuring that, and so it wasn't just North America. Australia. What they were doing, work they were doing in Australia for 60,000 years. Right when the white man first came, they couldn't ride their horses across Australia because the soil was too spongy. The horses' hooves were sinking into the soil because it was so spongy. Right, 10+% organic matter. No war in recorded history for 60,000 years. Aborigines were in Australia. They didn't have war. They're like, what's this war thing you speak of? Like, human nature, the essence of what humans can be and have been, I think is brilliant. And so I think we need to re-indigenize ourselves. It's much more about that sort of understanding to work with nature and listen to nature. And do we believe that communication can happen? Do we believe that little things come to our minds that are actually coming from nature? Is anybody okay with that? Can we talk about that? Some people? Because all indigenous cultures tell us that we can speak to nature and nature can speak to us. Right? We can speak to plants, they can speak to us. All those cultures who have a long track record are pretty clear. They get some really interesting information that would be hard to figure out without a PhD and a big fancy lab. They're like, oh yeah, we knew that. How the hell did you know it? Well, the plants told us. I think so. I think it's about a whole massive shift of our colonized mentality being